थैंक यू गोपाल जी सर।
गोपाल
जी सर,
मैडम को इंट्रोड्यूस करिए ना एक बार।
so Dr. Poonam Mishra madam is assistant professor at Kiran Patel University, Baroda.
Okay.
She did her PhD from IMTech Chandigarh and MSc from BHU, I think. है ना मैडम?
यस सर, यस।
तो मैडम कब किया आपने PhD?
सर मेरा PhD 2014 का है इमटेक से।
अच्छा, तब आप उसको जानती है अभिजीत बनिक को?
अभिषेक बनिक को तो अभिजीत भट्टाचार्य को एक जानते हैं, अभिजीत करके।
बनिक को नहीं? नहीं
बनिक को नहीं।
आप क्या थे? न्यूरो डिजेनरेटिव पे काम करते थे?
नहीं सर, न्यूरो डिजेनरेटिव पे तो काम हमारे यहाँ सच बाद में भले ही चालू हुआ, बट जिस समय मेरा PhD चल रहा था उस समय तो न्यूरो डिजेनरेशन की लैब नहीं थी कोई भी ऐसा।
अच्छा।
मैडम भी ली थी बायोइन्फॉर्मेटिक्स पर में लेक्चर, है ना? सेकंड लेक्चर मैडम का ही था इस साइंस डायलॉग सीरीज में। एंड देन मैडम इज आल्सो कंडक्टिंग दिस बायोइन्फॉर्मेटिक्स बेसिक बायोइन्फॉर्मेटिक्स कोर्स, जिसमें कि 135 पार्टिसिपेंट्स पहला बैच में थे। अभी सेकंड बैच 25 से शुरू होगा, है ना?
अच्छा
कोर्स।
यस सर।
सो हम लोग 1.5 साल से आए कनेक्ट में। मैडम का देखा पोटेंशियल बहुत अच्छा है। शी इज हैंडलिंग स्टूडेंट एंड पार्टिसिपेंट्स वेरी वेल।
थैंक यू
यस।
चलिए। Okay, so thank you Gopal ji sir, and I welcome Dr. Neeraj Mishra sir from the Gitam University to talk him about, since I have also listened or I have also attended the yesterday's session which was on sodium potassium ATPase protein role of in epigenetics. Now today what sir is going to discuss with us is the role of epigenetic reader proteins in health and disease. I think that this topic is more relevant, sir, to let you know that I have also worked in my PhD on the prion proteins. We have identified one prion in yeast system. So this topic actually it was really very intriguing for me to listen you about the topic after so much time. So I expect that this will be also interesting to all the participants, and to talk about the experience and the background of the sir. So he has completed his MSc from the Agra University in Department of Biochemistry, followed by his PhD from the School of Biotechnology, alumnus of JNU, New Delhi. Then he carried out different few postdocs, one at the Weizmann Institute of Science, Israel, one of the leading institute of the research occurring in across the whole world, and then to the Department of Chemistry and Medicinal Chemistry at the University of Minnesota. And currently sir is located at Gitam University in Vishakhapatnam since 2021, and also he is conducting research in the field of the epigenetic proteins and the regulation of human sodium potassium ATPase mediated by FXYD protein domains, and sir has also talked about it in yesterday's session. So I request Mishra sir, he continue the today's session and enlighten us with the topic that he is going to discuss with us. So sir, now the stage is yours and we are waiting for your presentation. So
thank you madam. Okay. So this session is interactive, so anybody can stop me anywhere, anytime, and any question or any confusion you have, just stop me. I will try to explain. Believe me, I will try my best. I am not guaranteeing you that I will answer all 100%, but I will try my best. So the topic of this talk or presentation is epigenetic reader proteins in health and disease. So that's a group of epigenetic reader proteins, but I will be focusing mostly on bromodomains. Why this topic is important? Why am I going to present? Again, yes, as I told you yesterday also, that I am interested in weak interactions in nature. So these all epigenetic reader proteins, they are mostly protein-protein interactions. Mostly, I'm not talking about all of them, mostly protein-protein interaction. They are actually weak interactions. They are not very strong interactions. So I will start from what are the weak interactions and which interaction you can categorize into weak interactions, moderate or strong interactions. So we will talk from there. Okay, so I will start from what are the weak interactions and why are they important and so on. Before that, I will tell about the security and safety of your lab and your department. So I'm sure that everybody has their pressurized gas cylinders in their lab or in their department. So how to store them? Usually, this is the way that we in India people store cylinders. You know, this is not safe. It is very dangerous because every cylinder here you see, this is a type of a missile. How does it do? This was somewhere in India, in North India, and you can see how it was done. Sorry, it should be video. I don't know why it's not working. Okay, sorry, so I cannot see. What happens? One of the cylinder, this guy, you say, he's taking out from his truck, and when he is trying to move here, so what happens? The cylinder falls down, and you see every cylinder has a very weak neck here. Like you see my presenter, right, Gopalji?
Yes, sir. Chal raha hai.
So you see that every cylinder has this top releasing gas place. So this is weak place. So when it falls, this place, this nozzle was broken, and the cylinder just ran out like a missile. So it's a lethal. It can break whatever it comes in on the way. So never ever store gas cylinders like this. This is the bad way. So how to store them? First of all, you fasten them. You restrain them with the wall or any immobile, strong, sturdy place, and followed by, if it is not in use, keep the cap. Okay? So this is the norm that you have to store your gas cylinder and keep safe everybody around you. So what happens if you look at the cell? Whatever we talk about, whatever we do, we always talk about the cell sap, cell inside whatever cell we have, like cytoplasm. If we talk about that, cytoplasm, we think that it is just a test tube and you can add, so it can mix. No, it is not. Cell has a very organized structure and very crowded structure. So whatever you see here, these pictures, these two pictures, this one and this one, they were made by Professor David Goodsell. If you want these high quality images and high resolution, you can download them from rcsv.org. These are free and you can download them at high resolution and you can print them. So you can see from here, cell is very organized. It's not a sack of the protein or whatever. You just keep every molecule inside in a bag and you can shake it and you have cell. No, it is not. So if you have any information, this starts from cell signaling right from the cell surface and it reaches to the nucleus right here. How does it reach? It has a very specialized pathway and protein-protein interaction. Whatever you see the down right here, this is the recent picture. I would not say that recent, it is around 4 years old. This is the most detailed picture that has been produced so far for the cell. So how signal reaches from upstream to downstream without moving much. You can see if I try to take protein from here to here, it will take very long time, but it doesn't take. For example, if you do any experiment, you add any molecule on the top of the cell and it reaches immediately to the nucleus, or it signal just propagate like anything very fast. How does it happen? The most important thing is it includes protein-protein interaction. How these protein-protein interactions are varied and how do they move the information from upstream to the downstream till to the end of nucleus. So these all are information are moved by information. Those are known as post-translational modifications. those post-translational modifications are like information or like a message, those are read by some other targeted molecules. So molecular interactions, how do they interact? You have a protein-protein interaction. We were talking about protein-DNA interaction, protein-RNA interaction, ligand interaction. Means you can have all those mismatch, mix and match type of interaction. So we will talk mostly about today protein-protein interactions. So protein-protein interactions, you can divide it into two categories. Number one is transient. Transient means for very short period, or you have a permanent interaction. How are they different? They are different on the basis of binding affinity, how these two proteins interacting with each other. So permanent interactions or long-term interactions, they have a binding affinity about the dissociation constant around the nanomolar range. Whereas transient interactions, they are also divided into two, weak transient and a strong transient. A strong transient means you have a shift from an unbound to weakly bound to the strongly bound state when triggered. So what happens? Do we have an interaction that binds very tightly but for a very short period of time? Then you have a weak in transient, he just comes, binds, and releases. Sometimes people are confused, is it a real interactions in weak transient, or it is a real interaction, or it's just non-specific interaction. So we will be focusing on this region, that is weak and transient interaction. So permanent interaction, this is strong. We were talking about KD is around micromolar, but it's a weak, then strong, weak, and strong. So typical energetics for non-covalent bonds. Yes, we were talking about whatever these interactions are. These all are, we are talking about non-covalent interactions. So typical energies for dissociation of the non-covalent bonds is salt bases, there are kilocal per mole, hydrogen bonds around 1 kilocalorie per mole, hydrophobic interactions even weaker, 0.7 kilocal per mole, whereas aromatic non-covalent interaction, that means pi-pi stacking, if I have two benzene rings, they ride on each other, this is around 1 to 3 kilocalorie per mole. So these are the bonds, whereas you have a covalent bonds, you can see they are much higher energy demanding if you want to break. For example, O bond in O2, it takes 30 kilocalories per mole when you want to break it. But if you go for an H bond, it takes around 94 kilocalorie. The strongest bond is CC bond. So look at the CC bond, it is 82 kilocalorie per mole. If you have a double bond, 147. If you take somewhere, it should be. No, it is not mentioned here. You have a CO bond. This is 164 kilocalorie per mole. So just imagine if you want to take out oxygen from carbon, from C double bond O, you have to invest 164 kilocalories per mole. But if you want to break any salt base, you have to just put 2 kilocalorie per mole. Aromatic 1 to 3 calories per mole. Hydrophobic 0.7 kilocalorie per mole. So you can imagine these bonds all together, non-covalent bonds together, they make a niche that is known as weak interaction. How are they important? So whenever you see there are two molecules are binding together, they have to have complementarity. So key of this universe, key of every reaction, key of the life or everything, whatever you see is complementarity. Until unless it is not complementing, you can't imagine there is something is going on. And also there are two things everybody has to remember in science: same charges repel, opposite charges attract. That's it. Science is finished, all reactions are finished. They start here, they finish here. Complementarity is the key. So look at here, you have OH and C double bond O. What do they form? They form hydrogen bond, because one of them is electron donor, another is hydrogen bond accept. Same negative and positive charges and so on. And here you see hydrophobic interactions. If it is hydrophobic and you put anything charge here, they will not interact, right? So complementarity is the key. How to maintain all this? So look at that. I will show you how are they making. So when you have a multiple types of interactions, Then you say this is a stable complex. It may not be a transient, maybe, but in this case, this side, it has a less stable complex, and definitely it is going to be a transient interaction. So this is a cartoon that was told. So why we are talking about these weak interactions? Weak interactions are essential for a life. If it is not there, it is not going to work. So ultrastructure of a cell is a consequence of executively non-random arrangements of the macromolecules that I told you in the previous slide. The set of strong interactions, 10 to 15 kilocalories per mole, decide the subcellular architecture element. So these strong interactions, they will decide how will cell look like, such as the membranes and the cytoskeleton, because they provide the skeletal or architecture to the cell. So they have to be strong. Whereas signaling pathways, signaling networks, and they rely on the weak and transient interactions. It's a fraction of few kilocalories per mole. So these are really weak interactions. All the interactions, whatever you see in the cell or in this universe, they are always under the evolutionary pressure. What is that under evolutionary pressure is whatever you are adding or taking out means you are changing the environment. So you change the environment, those interactions are going to be disturbed. For example, you are 10 people in a room and all 10 are interacting. You just take one people up or one person out, you left with 9. So whatever intensity or time you were spending, now you are spending with 9 people. So it's an evolutionary pressure. Or you just add one person, you are dividing that whole time and attention among the 11 people. So these are the evolutionary pressure. Same thing happening for the molecules inside the cell as well. And these evolutionary pressures are very important. They are essential for the fitness of the system, in other words, for the organism. In 1982, Professor McConkie, he coined a term quinary. What is quinary? He gave a definition for that term, and it is very important. I think everybody should learn it. If you are a student, think about it. The set of evolutionarily selected weak interactions that are essential to maintaining the functional organization of biological macromolecules in living cells. So weak, strong interactions, they make the structure of the cell, whereas the weak interactions, they move the cell. They move the functioning the cell. So, for example, whom do you like most? Who is your most nearest person, nearest people? That those people are your family. You are interacting with them very strongly. Without them, life is not easy. Without them. Then you have your friends. You have a friends. You need them. If they are not, you can live. But without family, it's very difficult. Without friends, you can live. But there is a third class also. For example, you have a plumber, right? You have an electricity problem, you have electrician, you have a plumbing problem, you have a plumbers. So when problems are there, call the plumbers. I'm sure that most of you don't remember who was the plumber. But he made sure that your drainage and pipes or everything is clean. If plumber is not there, you have a problem. If electrician is not there, you have a problem. But they are not the part of your life. You are just paying them, taking their services, and things are gone. So those plumbers are your weak and transient interactions inside the cell. Those are the life. The problem with them, how many of you know the plumbers or electrician who was in your house for last time? As long as he's not from your neighborhood, excuse me, I'm sure most of the people don't remember. So even the gentlest separation, excuse me, sorry. So to analyze those interactions, we have to separate them. We have to find them, what are those transient interactions. But the problem comes, the gentlest separation methods are capable of disrupting them. So when we try to study them, most of the time we lose them. So what happens if you take the overall cell it has been predicted the human complement of PPI, protein-protein interactions, numbers between 1,30,000 to the 600,000, means 6 lakhs. So it's a very large number. Weak and transient interaction are responsible for majority of biological processes. Weak and transient complexes are characterized by the fine balance between the specificity of the binding and the fast turnover rate. The majority of weak interactions display KD within the micromolar, even millimolar range. So whatever we talk about epigenetic reader proteins, protein-protein interaction, most of them are falling into micromolar range. Weak and transient complexes in biology are crucial for diverse biological processes such as biochemical pathways, signaling cascade, host pathogen interaction, and cell-cell communication. Majority of the protein-protein interaction involved in membrane transport, regulation, signaling transduction, and so on. So replication, transcription, translation, and many more. We will not talk about them, but it's okay. But their topology look like that. Cytochrome C, these are the 5 types of protein-protein interactions can be divided into, but all of them are protein-protein interaction in the week. So systems that I have studied so far and I will be studying in my lab, one of them I have presented yesterday itself. The latest one that I studied, that was the M tuberculosis aspartate pathway. And before that, I had this epigenetic reader protein interactions with the histone and sodium potassium ATPase. And the oldest one is protein co-solvent interaction. So this is the weakest, this is the weaker, this is also weaker, and this is the strongest among all. So I'm going to focus in my lab the sodium potassium ATPase and epigenetic reader proteins and their interactions with acetylated lysine containing proteins. So that those are known as bromodomains. So these are two my words. So as we know that every cell in our body has the same DNA, yet we have more than 300 types of the cell. How they have, and different cells do so many different things, even they all have the same DNA. How do they remember their fate? So for example, you have a muscle cell, when they divide, it makes muscle cell. It doesn't make fat cell. Fat cell makes fat cell, it doesn't make muscle cell. Bone cell, I'm talking about in normal circumstances. So how do they remember? They remember because they have a set of gene that is specific for immune cell. So that will be activated and that will be expressed and that will make cell immune cell. It will not activate the set of genes which is required for the fat cell. So, but since they have a same genome, they can be interchanged. They can interchange between any two types of the cells. But before that, you have to shut off. For example, you want to make immune cell to the fat cell, so you have to shut off the gene set which is required to be specifically for immune cell, and you have to activate the set of genes which is required for fat cell. So this process is known as induced pluripotent cell, right? Stem cell. So that is known as the stem cell. Pluripotency means you have pluripotent cell means you have a cell that can differentiate in any type of cell. So that is induced pluripotence. So how is it happening? Because you are not playing with genetic information. Are you playing with? You are playing with the information that is above the genes, but that is still inheritable from one cell to the next generation of the cell. So therefore it is known as epigenetics. Epi means on the top of the genetics, that's why it is known as epigenetics. So this is a new science, a new direction of the genetics. So we will talk about a little bit about the basics and the background of epigenetics. The term epigenetics was first introduced in 1942 by Conrad Weddington, then defined as a causal interaction between genes and the products that allow phenotypic expression. So this is the definition that Conrad Whittington gave. So the heritability of epigenetic information was for many years thought to be limited to the cellular divisions, but now it is now apparent that epigenetic processes can be transferred in organisms from one generation to another generation. But how? We are still struggling to understand, but there are few epigenetic marks such as methylation and acetylation. Those are considered to be carried out from one generation to another generation. So this phenomena was first described in the plants. Now it has been seen in yeast, Drosophila, maybe mouse, maybe possibly in humans. So mouse and humans, very few studies has been done on mouse. I think 2 or 3 studies came out about the humans also. But mechanism has not been completely understood yet. So epigenetics is the recent definition that is accepted by everyone. That is epigenetics is the collective heritable change in the phenotype due to the process that arises independent of primary DNA sequence. Means primary DNA sequence doesn't play any role here. So how does it happen? So those are factors for epigenetic regulation which are decided the fate of the role of the cell. So how many things you have that genes associated? You have a DNA and you have a protein. You have a 2-meter long DNA in every human cell. 2-meter long DNA is packed into some microns. Few micron cubic volume of the nucleus, it has to fit there. So how does it fit? It has several magnitude of organization. So you have a DNA, then DNA is negatively charged molecule because of phosphate all around it. If you put all DNA together, that I told you a few minutes before, that same charge is repelled. Unless you don't quench the negative charge, there is no way that you can put all the DNA together. So you have to quench the negative charge and make it neutral. How can you make it neutral? You cannot put more positive charge because then again it will repel. So how will you quench that? You have to find out some binding partners. They are binding with negative charge and neutralize the negative charge. So what do you need? You need proteins. Those are the easiest not easiest. Those are the molecules. Those are going to bind with negative charge of DNA, make them neutral, and DNA wraps around them. So those are the proteins known as histones. Those are highly positively charged proteins. They make it, those are the nucleosomes. So you have 8 protein molecules. You have histones, are 4 types of histones. Number 1 is H2A, second one H2B, third one is H3, and fourth one is H4. So this is the core of the nucleosome. These every histone, H2A, H2B, H3, and H4, they have 2 molecules of each. So 2 molecules of each histones of 4 types. So that means you have 8 molecules. They form a coin-like structure and DNA wraps around. And this structure is around 11 nanometers wide and known as V-Don string. Then it's further condensed with the help of other proteins that includes H1 histone, and that is known as 30 nanometer chromatin. You have again keep adding other proteins and you condense them. So what do you think? You have, in the end, you make a chromosome. That is around 1,400 nanometer wide. So from DNA to the chromosome, the DNA is packed. Now DNA is packed until unless DNA is not in this form, it is not usable. So in order to express any gene or any part of the DNA, you have to remove from here and make this region. How do we make it? Remove those histones. How can we remove those histones? We will talk about that. So here you have a histones and you have a DNA, and both molecules can be modified. You have a DNA methylation and you have a histone modifications as well. So these all together, modification of DNA, chemical modification of DNA and chemical modifications of histones, those combined together, those modifications, they regulate the set of gene expression. And that is heritable to the next generation of the cell, next cell division, because it is not related to the gene, it's related to the modification of the DNA and the histones, therefore it is known as epigenetic. So what are those? You have histone acetylation, you have histone methylation, and non-coding RNA and DNA methylation. So these 4 types of epigenetic factors you have. Not 4 types, I will say histone modification and DNA methylation and non-coding RNA. So 3 major types of. So we will talk about histones only right now. So you have a histone gets post-translationally modified. Look at the complexity. I want you, everyone, if you are a student, think about the complexity of this histone code. It is also known as histone code or epigenetic code given by Professor David Ellis. He died last year. Otherwise, he could have a Nobel Prize. So there are more than 70 sites in all 4 histone molecules. Not 4, sorry, 5. You have H1 also. So all those 5 histones carries more than 70 sites which can be chemically modified. And most of them are at unstructured N-terminus tails of histones. So 70 sites and more than 15 types of chemical modifications has been identified by 2019. This number is around 19 now. So amino acids, those are modified, or you can see here, you have a lysine, arginine, serine, threonine, tyrosine, histine, and this is, I think, glutamate. So all these amino acids can be modified, and one of them, for example, lysine can be modified, acetylated, can be methylated. Even 3 types of methylation occurs for lysine. So lysine itself can modify it for 4 types. Arginine can modify it more than 1. Serine also, and threonine also. So you can see there are lots of mix and match. If you see the numbers of possibilities, how many are these? Can create, that is more than genetic variability you can have by DNA itself. So it is more complicated than your genetic variation. So most of, not only that, let's add one more complexity on that one. That is also very common. So most of these post-translational modifications are not present alone. They are present in groups. So for example, you have a lysine that is acetylated. Same time you have a serine that is phosphorylated. You have again histidine that is also phosphorylated. So there are, and lysine and arginine, they are also crotinylated and so on. So these one histone can modify multiple sites and multiple variations. So now you can imagine multiple sites, multiple modifications, and on multiple histones. So extremely complicated. And those modifications are upstream signaling specific. And those modifications are responsible for release of DNA from the chromatin or bind or forming the chromatin out of the DNA and histone. So this is the reversible. So all these post-translational modifications are reversible and dynamic. So you can imagine how difficult it is. So this is a snapshot published in 2014 in Cell, and you can see these all are the type of modifications were recognized at that time, from very small from acetylation until here you have big nucleotides attachment to the histone site. And you can see one site here you can see it can modify it several times, here and so on. So you can now try to mix and match and see there are enormous and countless possibilities. So because of that, David Ellis gave the name histone code and epigenetic code. So this was the only about histone. Now you think about, you have also DNA that can get modified. So all these together, DNA plus histones, it's extremely complicated. So therefore, it is known as epigenetic code. So I found this picture and it was really nice. I think I should have studied something easier like quantum.
Poonam madam, you have raised hand. Do you have any question?
Yes, sir. Sir, in the previous slide, I just want to ask.
This one?
Yes, sir. You have mentioned that there are many post-translation PTMs are possible at the single amino acid of the protein. So
yeah, you can see.
Yes, sir. The single site that you have shown here, then how it is possible for the cell to determine that which kind of PTM has to occur in a given condition?
So, madam, that is, again, I told you this is, I didn't tell you, this is a very new field. So whatever upstream signal is coming, that will induce the enzyme or induce some other protein that will deposit this mark. So these marks are deposited by writer proteins. That will become next. So all those markers, those deposit writers, proteins are, for example, acetylase. Those are known as histone acetylase, DNA methylase. So those all enzymes has to activate it. How do they can get activated? Some of them get phosphorylated, some of them get dephosphorylated, some of them binds with some sugars, some of them binds with some lipid. So all these modifications also gets, they activate the proteins. Those are enzymes, those are going to deposit these marks. So this, madam, depends upon the what is your upstream signal that decides.
Okay.
नहीं, मुझे लग रहा है मैडम जो क्वेश्चन पूछी है कि हाउ सेल डिस्टिंग्विश की सपोज सेरीन पर होना है ना फॉस्फोराइलेशन और दूसरी जगह भी सेरीन है सो देयर एडजेसेंट अमीनो एसिड रेसिड्यू विल गिव सम सिग्नल ना दैट इज डिफरेंट
यस यही ओके
आसपास का जो रेसिड्यू है दैट इज एक्टिंग एज अ सिग्नेचर ना
एक्चुअली दिस इज बिकॉज द So what happens? Every enzyme has a different specificity, right? So those specificity are determined by, again, what I told you, this is about the complementarity. Until unless it doesn't bind properly, it will not react. So the adjacent residues, not only adjacent residue on one protein, because these are the group of proteins, so all proteins together, they will also decide which protein can bind and fit here. So all together, this is no structural complementarity will decide which protein and which residue is going to be modified.
Yes, yes.
So we have to find out. Actually, we are still in dark.
Sir, this is also a kind of conditional, the things which are occurring based on the condition, situations.
Definitely yes. So for example,
based on the outside signal.
Yes. So for example, if you are giving somebody glucose, right?
Yeah.
You activate glucose transporter in the cell, right? And it comes to the end, it takes glucose. If you give more and more glucose, what happens? It same molecule, same transporter. Now you have turning glucose into glycogen first. If it is not even enough, then it will start making other enzymes. Those will make it. First, it will make pyruvate. Then they will make fat. So one molecule and one molecule is activating several different molecules on the basis of its concentration, right?
Yes.
So this is all about because some molecules have a high affinity towards that particular molecule, some other protein molecules have a low affinity. That's why it is getting distinguished.
Okay, sir. Okay.
So how these marks are deposited, read, and erased that we talked about, these are reversible. So first they deposited and the response of the upstream signal, then they were read by the appropriate proteins, followed by the erasing that mark. If you don't erase, the reaction keep going. So You have writers of the proteins, they histone acetyltransferase, protein methyltransferases, and so on. Then you have erasers, histone deacetylase, also known as HDACs, and these are DACs. Then you have readers, the recognition of the proteins. We will talk about them in a minute. So first you deposited marks. There were several, so you have a different reader of the proteins. So arginine, methylated, that's a recognized methylation, recognized by several types of the proteins. Look at these. These are all the group of proteins. They recognize methylation. But acetylation of lysine is recognized by mostly bromodomain. Those proteins which recognize acetyl lysine, those are known as bromodomains. So my focus of study is bromodomains. So we will talk about them. So what are the bromodomains? So everybody must not confuse bromodomain means bromine. There is no bromine in bromodomains. So bromodomains in human, there are 46 proteins, and 46 proteins contain 71 bromodomains. So that means some proteins, they have a multiple bromodomains, such as this one, you have a PB1, you have a polybromodomain. It has around 6 bromodomains. Then you have here this protein. This protein BPTF most probably, so it has a 1 bromodomain. Crab binding protein, very important protein, it has only 1 bromodomain. And the most studied group of bromodomain is BAT, bromodomain extra terminus proteins, BAT bromodomains. So they have 2 of them. So there are multiple. So reason is, look at that. These are the big proteins, bromodomains are very small. So they are the part of big proteins. They are the domains of big proteins. So they recognize the histone or protein modifications, acetylation, and rest of the protein work. So these are kind of anchors. So they recognize and they bound. Now you think about it. All bromodomains, they recognize acetylated lysines. You have a 46 proteins, they have a 71. You think about it, why nature needs 71 bromodomains? Madam, I'm trying to answer your question. Why do you need 71 bromodomains? Because all 71 bromodomains have a different affinity towards the histone modification or acetylated lysine. And that is strengthened or weakened by the additional modification of the chromosome. So therefore, they have a very specific interaction partners. So if you divide them, so they can be divided into 8 groups. So if you look at here, BRD2, BRD3, BRD4, and BRD3, this is your back bromodomains. You have this third part, CREB-BP. And I think there is a very important one. This is a PAL-G. This is also known as BPTF. And GCN5. These are most of them are transcription factors. Now, if you think about it, why transcription factor needs bromodomain? Because cell has deposited acetylation mark on particular gene or particular promoter. And that has to be recognized. So that will be recognized by some specific transcription factor in order to start transcription. Some question is there?
नहीं सर हमने वो पेस्ट किया है अपना सोशल साइट का.
Okay.
We will keep in touch, है ना?
Okay. So my lab is also interested in POL-G. That is also BPTF, and this one, CREB-BP. So these 2 proteins we are interested in our lab. Why? Because these proteins are the least studied. There is one more protein, ATAD2. It should be somewhere here. I don't remember exactly which group it is, but that is also very important. BRDT, BRD4. So these are the proteins that I have studied and I will be talking about today. All right? So, and they are, now since they are expressing and controlling the expression of the genes, so you can imagine They are involved everywhere. Whatever you think of the cell fate, they are there. Chromodomains are there. So they are involved in disease-like situations. So you just mess up with them, you overexpress or downregulate any gene product, you have a disease condition. So bromodomains in diseases, inflammation, cancer, cardiovascular disease, obesity, neurophysiological disorders, neurodegenerative diseases also, viral diseases. You can imagine even viral diseases also. Eukaryotic parasite and the diabetes. So what happens? Whatever you see here, these three highlighted one, I have worked on these three systems about of bromodomain of three the model systems. So what happens? So what are they doing? They are just recruiting transcription factors or recruiting other proteins for further process, such as a transcription, translation, or replication. So if it is going out of the way, if it is not following the proper rules, what do? You just stop it. You just stop it, then probably cell will forget what is it. So what happens? Then in 2010, this paper was published in Nature by James Brander and Stephen Knapp. And this was the biggest hit. Before that, it was very difficult to target these proteins because these are the protein-protein interaction and their surface or binding pocket is very large. So they were known as undruggable. So what did they do? This guy, he designed one molecule and synthesized, and he showed it that you have a cancer cell and you treat with that molecule known as JQ. It's his scholar's name, GenQ, and you keep adding. You can see after 72 hours, cell forgot that it is a cancer cell, and cell start behaving like a normal cell. So it was a very big hit, and everybody wants that molecule. But later on, they find out the cell developed resistance against this molecule also. So it was hit, then flop. Anyway, so this is the JQ molecule, and it inhibits back bromodomains. It does not distinguish between BRD4, BRD2, 3, and T. So these are the bad bromodomains that inhibit for the midline carcinoma, spermatogenesis. So here, this is the common structure of bromodomain. And you see this magenta, this is your peptide, this histone peptide, and this is acetylated lysines, they bind to this pocket. So if you look, this type of any arrangements, how will you study this? Because this is not enzyme. So you have to have a readout. So the readout that is most common is you have these peptides, they are fluorescent tagged, and that's I'm talking about high throughput. You have to screen many molecules to break this interaction out. So you have this fluorophore bound, then you check fluorescence polarization. When they bound, you increase the polarization. When they are free, you have a less polarizability. So in that case, bound form has a more polarization, and free form has a bound form has a more polarization, free form has a less polarization. So the window of fluorescence polarization was used for study those molecules, but it's not easy because you have a 71 bromodomains, and for that you don't know which histone modification is perfect. So that was the difficult. That was the only way what happened. So in the lab I was working as a postdoc, they come up with the idea, okay, let's modify protein itself, and we do it. So these are the inhibitors that people were talking about. They have against these molecules, and how to find out? So protein-protein interaction, I will not talk about that, so leave it. So if you study what are these here, you have a small protein that is histone, and you have a bromodomains. This is also part of protein. This is also protein. So you have a protein-protein interaction. Now, what is making them interact? That place that is going to tell you known as hotspot. So those hotspots were analyzed and found out that they have aromatic amino acids. They are rich in aromatic amino acids. Mostly, if protein is going to bound to another protein, in most of the cases you will find tryptophan there. If you have a tryptophan, it is means on surface that probably you are going to bind something.
Chaitanya, do you have any question? You have raised hand. You may unmute and speak, or you can write in chat box. Chaitanya,
Yeah,
by mistake you have raised hand.
Okay. Any question? So by mapping those hotspots, if you have any protein structure, you just go and see in PDV, and you can think probably some protein is going to bind there. So that is known as hotspot identification. This paper was published in Nature in 2007, and it was so successful because most of the protein which are interacting, they have hotspots. In bromodomains and histones also you can see that is aromatic rich region. So by looking at that, we realized, okay, so let's see. So we find out few bromodomains, and you can see they have a WPF cell. This is known as WPF cell, and this is common. You have this tyrosine is also conserved, this tyrosine is also conserved, and this phenylalanine, and this tyrosine. So you can see that's a big numbers, not big, that's a number of amino acids, those are conserved, and most of them are aromatic amino acids. How can we exploit them in our favor? So what did we do? If you map these in BRD4, you can see this is the place, they are here. This is the JQ1 that is bound to the active site, and you can see these amino acids are aromatic amino acids. They are involved in protein-protein interaction. Can we use these aromatic amino acids in our favor? We will see how can we do that. So this acetylated lysine binds it. So you have a tyrosine 139. This is about bromodomain BRD4. We are talking about BD1. And look, in 97% of bromodomains, this tyrosine is conserved. This tyrosine is conserved in 92% of bromodomains. So that means we can exploit these residues in our favor as a signal producing residue. So how can we use that? Because tyrosine does not have a very high fluorescence, and protein has a multiple tyrosine. Protein has a multiple tryptophans. This is only conserved in 81%. So we can exploit only these. So let's do what can we do. So what if we label these side chains for some nuclei or some atom which is responsive? So what did we think about it? So let's put fluorine on them. So if you make this a tyrosine, make fluorotyrosine, what will happen? That is an NMR active nuclei. So if it is there and anything binds here, you will see the shift in chemical shift. That will be changed. So let's do that. So why fluorine? Because fluorine is hyper-responsive chemical shift. It is 100% natural in abundance. Do you know that in no natural molecule has a fluorine? So if you see any fluorine resonance, that means that is incorporated by non-natural means. Then you have a high signal sensitivity. So it's around 87% of the proton NMR, proton itself, and low number of signals since you don't have a background noise. So let's introduce fluorine in our amino acids. So how can we do that? The reason why are we doing? Because those protein-protein interaction study is not easy because it is not having enzymatic activity. It does not have any direct readout. So first of all, to crack that bottleneck, we have to come up with some idea that we can use it. So we came up, okay, let's do protein nozzle fluorine NMR. In short, PROF. So how do we do that? So you take bacteria, insert your plasmid of your desire, whatever you want to express in the protein. Take the bacteria, grow in LV, let them grow, then remove all the amino acids. Remove the amino acids and use the defined media. Instead of giving them normal tryptophan and tyrosine, if you whatever amino acid you want to, label with fluorine, give that amino acid with fluorine. So, for example, in this case, we are taking out either tryptophan or tyrosine. So we will replace this tryptophan with 5-fluorotryptophan, and we will replace tyrosine by 3-fluorotyrosine. So you have a defined media that has all 20 amino acids and nucleotides and the carbon source and so on, and your desired amino acid. But problem comes here again, because bacteria can synthesize its own. Since these amino acids are not natural, so bacteria will synthesize its own. So what to do? You have to shut off, or you have to stop the biosynthetic pathway for these aromatic amino acids. And that pathway is known as shikimate pathway. So you stop the shikimate pathway, so bacteria will not be able to make its own aromatic amino acid. Now you can feed them. So now you will make bacteria to eat whatever you are giving. So for example, if I want somebody to feed salad, I'm sure that most of the student doesn't like. How will I make them to eat salad? Keep them starved. If they are starved, they will eat salad. So same thing happens. We keep bacteria starved with that amino acid. It has to take. Right? So at the place of natural tyrosine or tryptophan, bacteria start using 3-fluorotyrosine or 5-fluorotryptophan. Yes, these amino acids are toxic, so you have to be careful. You cannot feed more. You have to be very careful. So don't put much. You don't need to. So if you express wild type protein or wild type BRD4, you can express in large amount. You have 120 milligrams per liter. Now, in BRD4, you have a 7 tyrosine. 7 tyrosine means you have to add lots of fluorotyrosine. Since it's a toxic, so protein yield gets low. And a bacteria grows also slower. You get around 25 milligrams per liter. That's not bad, but it's not great either if you compare with the wild type. Then you have a 5-fluoroindole. You have only 3 of them, so therefore you have a jump. The yield, it's around 88. Then you have another protein, BPTF, that goes around 84. That is enough for 246 NMR experiment. Means you can do 246 NMR experiments by using only 1 liter medium. So not very expensive, easy method, but you have to have a resource. That means you have to have an NMR machine. Then only you can do, otherwise you can't. So if you want to study this, how can we do that? You go and read this nature methods, G et al. This is my paper also. And you also follow this Mishra et al. That was published. Okay. So when you take, this is BRD4. This is 1D NMR, but taken in only 10 minutes. So you take NMR tube, put around 40 micromolar or 50 micromolar of the protein, and you see you have all those residues are here. All the resonances 1, 2, 3, 4, 5, 6, 7. All those residues are well separated, well dispersed. All 7 fluoro you see. But how do I know this is belong to bi-97? This is tyrosine 137, tyrosine 65. How can you do that? You have to assign those resonances by mutation. So just take this Y97, change Y to the F, means change tyrosine to phenylalanine. In that case, this resonance will be eliminated. You want to see where is Y137? So mutate this one, and you will see this resonance is disappear. So by removing or site-directed mutagenesis, you assign. Those are the fluorine resonances. In case of tryptophan also, you can do the same. There is another way. For example, I don't want to play with this all. I don't want all the tyrosines are level. I want to level only this one, 137. How can we do that? There is a method that you can do that is known as genetic code expansion method. that you can selectively label wherever you want. So since we have a protein that was native, is it working or not? We don't know, because you have a loaded 7 fluorine, and fluorine is very electronegative, isn't it? So fluorine can crash the protein, it can aggregate the protein. Is it worth working or not? So you have to check it. How can you check it? You have a structural determination, as well as you have functional determination. So what did we do? How do we know what is the protein is going on? So we take CD. So we took CD, circular dichroism, and we find out that you have almost the same secondary structure in all of them. Wild type of BRD4, 3-fluorotyrosine, labeled BRD4, and 5-fluorotryptophan label. They have almost same secondary structure. And are they stable or not? So the next thing is, let's melt them, heat them, and you calculate the melting temperature, and you can find out all those three have a similar melting temperature. And to be confirm and sure, let's do some structural analysis, and we crystallize and we overlay with the labeled and non-labeled, and you can see they are overlapped, so they are matching everything. So this is the structural part. Okay, is it going to bind with our protein or not? So you can see right here, to analyze the gold standard, and gold standard technique is ITC, isothermal titration calorimeter. You can see they both bind exactly with the similar KD. This is wild type, this one is your 3FY, and this one is your 5FW. So they don't change much except 3-fluorotyrosine level, because you have a 7 fluorine there. So by doing all these experiments, we proved, okay, it doesn't change secondary structure, it doesn't change stability much. Yes, tyrosine labeling changes protein little bit, but can we use it? Yes, we can use it. So this compound was known that it binds to BRD4, so we synthesized in the lab, and it's a very tight binder. Some you can say this is how much? It's a tight binder, right? We were talking about this is 75 nanomolar affinity. So when you add it, so NMR is also good, because if you have tight binders, the resonance just disappears and it starts showing another place. If it is weak binder, this resonance or any resonance will start shifting, either left side or right side. So what happens? You add, this is your no JQ1, this molecule, there is no, then you added 0.5 equivalent, means the half molecules of JQ1 compared to the protein itself. So you can see, you assume around 30% or 25% protein is bound, and rest of it is free. Because it's a tight bounder, you can see this very tiny resonance is appearing. And this resonance reduces the intensity, isn't it? Now, okay, so let's jump to the 2 equivalent, means you want every protein molecule to be engaged. And you can see this resonance at W81 is completely disappeared, and you have a new resonance. What does it tell you? It tells you that this resonance, this amino acid residue is directly engaged with the binding of the ligand. Where is it? You can see it right here. This tryptophan W1, it present at the active site, and it's directly engaging with the binding of histones. Now we have a system. Now we know how is it going to work. Can we use it for a screening or some other thing? Yes, let's see it. So this one was the tight bounder. What about the weak binder? So there is a molecule known as acetaminophen. Can anybody tell me what is that? Acetaminophen? Acetaminophen. Can anybody tell what is this molecule?
It's a
Painkiller. Painkiller.
It's a painkiller.
This is the painkiller, paracetamol.
Yes, sir.
So this paracetamol also binds with bromodomain. There was only one study That was the crystal structure that paracetamol or acetaminophen binds to the bromo BRD4, BD2, second bromodomain, at very weak interaction. Only crystal structure could see that, no other method could see. Can we detect with this NMR method? So what did we do? We took for JQ1, that this is a previous that I showed you the structure. Now let's start adding acetaminophen. And you can see, so this is a weak binder. You can see this is the...
Sir, how much time required? More time?
No, you don't require more time. So one, you see one spectra, it's a 10 minutes. We have taken around 5 minutes also. It was not that clean, but it was good, since your resonances are clean. So you can see this is a weak binder. You can see the shift, right? The left shift. And here, when you take the tyrosine spectra, I cut few resonances from here because I'm interested here. So this is in wild type, so without any acetaminophen, you can see this resonance starts shifting this way. So that means we can pick very weak binders. Since it's a weak binder, we can draw and we can calculate the KD. And you can say Y39 dependent KD was 290 micromolar. That means 0.29 millimolar. I'm sure that it's a very, very difficult to capture this interaction by any other method that you could capture by fluorine NMR. Similar numbers also you get when you have a W81. So that means you can use this method to find out the strong binder and the weak binder. So method is proved, and we published that in ACS Chemical Biology. Now can we use this? So remember that if you have two different molecules, the resonance don't affect each other until unless they are not having J coupling or other methods. Means they are not coming closer, they are not interacting directly. You don't see the shift in chemical shift. So what do we do? For example, you have 2 or 3 more proteins. If they are not interacting with each other, you can use them you can use them all together. So more than one protein can also be used if their resonance are not overlap. So in case of that, we took BPTF bromodomain and we took bromodomain BRD4. So we had a BRD4 labeled with a tryptophan, and here we have a BPTF tryptophan label. So what did we do? So take this. Remember that you have a BRD4 and BPTF, both proteins in one NMR tube. And you can see these 3 resonances are from BRD4 and only this one. This one is from BPTF. Now these all resonances are not overlapped, both proteins showing their different resonances. If I'm adding any molecule, if it is specific for one protein, right, then only one protein's resonance will move, other will not move. So by using this method, we can also determine the specificity of the molecule. So you can add more than one protein and add molecule and see. So this is kind of a first. What do? We take molecules, mix them, and add in one protein and see whether it's affecting or not. Then we deconvolute. So that's a screening method. So in this case, what did we do? We took 2 proteins and we started screening. So we bought a GSK library. This was very small library. This PhD student was very lucky, I would say. Luckiest person I have seen in my life. So he took 229 compounds. That is a protein kinase inhibitor library from GSK. And he did protein observed fluorine NMR. He found that 9 molecules, they were hitting BPTF, 31 molecules bound to bromodomain, and out of 31, 15 were selective to the bromodomain, the BRD4, and out of these 9 hits, 3 were selective for BPTF. Again, you found those hits, you have to deconvolute again, you have to discover whether they are specific or not. When we found that one molecule that was specific for BPTF bromodomain that was not binding with any bromodomain or BRD4 or other bromodomains we had. So what did we do? We took this molecule AU1, it seems here. This was the molecule that we found. This method is known as fragment screening. Why do we call it fragment? Because this is a small molecule. Actually, this seems a larger molecule compared to the fragment, but I don't know why it was there. But this is a fragment. Look at this is a big molecule. We find out, and it was moving only tryptophan residue, that is 28, 24, and you can see. So by titrating this, we also find out what is the binding affinity, and this was the case. So we confirmed that with the ITC again, and you can see it was binding to the BPTF bromodomain around 2.8 micromolar. So this is the molecule, AU1. Look at that. So next step comes when you have these molecules, how will you make use of these? Because this is not very good molecule. Only good thing about it, this is a selective molecule. So what did we tried to do several things. We deconstruct this molecule and reconstruct, but no success. So that was a dead end. Whether this molecule is important, can be used in cell or not, we have to finish the story. So what did we do? So we find out, we looked into the literature that which disease cell or what disease condition this protein BPTF is going overexpressed, right? So what do we need that? So that I told you before, if you take if you see the bromodomains, they are the small part of the big proteins. So if you knock down the proteins by siRNA or any other mean, what do you see? You see the effect of the whole protein. If I have a specific inhibitor that binds to the bromodomain only of that protein, that means I am neutralizing the effect of bromodomain. The rest of the protein is there. So therefore, we have to have very specific inhibitors. So what is going? When we realized, when we studied and looked around the literature, and we find out in breast cancer, BPTF is overexpressed. We again dig deeper and we find out it also expressed in cardiac hypertrophy. Okay, now we have a system. So which one is in our reach? And we find out the breast cancer one was easiest one. You can grow the cell lines. There are lots of groups are working. So we sent this molecule to another group in Cold Spring Harbor Laboratory, and we checked whether it is going to work or not. By all means, there was no toxicity until 10 micromolar, and it was affecting the cell growth of the cancers. So that means this molecule was great, not great, it was okay. We had several attempts. Finally, we couldn't do that. By using this propanema, there was another molecule, but that was another story. I'm not going to tell about that because I was partially involved. But in this study, I was started completely from the beginning. But of course, it happens always. You have to give your project to PhD students. So I gave, and finally, we have that molecule, and it was published again in ACS Chemical Biology. And this is one of the highest cited papers. Thank you. So right from the developing a system or a method to have one molecule, that was the success, and we did all this work within 2.5 years. So you can see that it was a good work we did. So I'm done with that.
Thank you, sir. Poonam madam, and any participants have any question? No question? बहुत कम क्वेश्चन पूछा आज लोगों ने, पार्टिसिपेंट्स ने. प्रोटीन इंट्रैक्शन सबको नहीं समझ में आ रहा है ऐसा. दिस इज वेरी सो हमने भी पीएचडी में बहुत सारे कोइमिनो प्रेसिपिटेशन, पुल डाउन, को एक्सप्रेशन ये सब किया था. आईटीसी नहीं किया था, सीडी थोड़ा सा किया था. To understand the architecture and assembly of type 4 secretion system of Helicobacter pylori,
E6
हाँ, type 4 secretions, yes. So उस टाइम में बहुत भारी लग रहा था, कुछ किया, but ये बाकी सब नहीं किया था. So it's good.
It was a big problem because if you don't have a method, how can you How can you do?
And
So
handling is also in precipitation and pull down. Handling is very much.
Gopalji, if it is weak interaction, you are not going to pull at any point.
Transient interaction through pull down we cannot see.
No, it's not possible because
Yes.
you increase salt concentration, you decrease salt concentration, it is gone.
Yes.
So you have to be. No, there is no way that I can tell you that.
Cross linking करके देखा जाता है. Sometimes cross linking.
No, you can do that, but that's not specific. How do you know this is directly binding or not?
Yes. So sometimes bridging protein plays a role,
Exactly. So
and
that is
pull down we cannot determine.
You say bridging protein in the biophysical chemistry we call it ternary complex.
Yes.
So if it is a ternary complex or binary, usually we are interested in binary, right?
Yes. So जितने भी signaling pathway है, जितने भी जो complex protein है उसके लिए all these techniques are very much essential.
Very essential.
Yes. Now,
let me tell you one thing about that. So, for example, I incorporated fluorinated, right? By using another method that is also known as genetic code expansion. First of all, because tyrosines are all around, you fluorinate them and add in cell lysate, you will find out which resonance is moving, right? Then you take that position and you incorporate any photo cross linker. And in normal cell and disease cell, you capture that. So that will be your differential interaction can be also captured transient. So that is the next level of your determination, understanding of protein-protein interaction. That what we can do.
Yes.
So that next step we are doing in our lab now.
Yes, sir. Actually, sir, I have also done a multi-protein complex study. One protein with chromodomain protein.
Chromodomain, yeah. So it binds with the
chromodomain protein. One protein in yeast that was a part of anaphase promoting complex. Multiple interactions were there, and
crotonylation is detected by the chromodomain.
Yeah. And one of the major protein which take parts in formation of heterochromatin, the trimethylation, chromodomain proteins are there. So we also, as Gopalji sir was saying now, that for co-IP and pull-down experiment, if the weak interactions are there, we also found out that ubiquitination, that was monoubiquitination was required for this kind of interaction for among the different partners of those anaphase promoting complex. So a bit the talk that you have given to today, that was really kind of rebrushing the things, because I could correlate few things and a bit of doubt that were there in my mind regarding the post-translation modification at the, as you told, that many can get across in the same protein. So I really appreciate, sir, that this talk was really helpful for me. And I hope for rest of the participants also, they might have enjoyed it. So it was really nice one.
Thank you, madam.
They can share their feedback. Yes, sir.
Yes, sir. Namaste, sir.
Namaste, sir. It was very, very interesting. I listened to your lecture in a rapt attention, because this is not my much area, but since we keep on hearing from colleagues who are working on chromatin modifications, many of these terms are very much familiar, and I was just trying to make out all the links, and I really enjoyed this very much, and this was also you have worked in a very professional way to identify the interaction of the molecules, devised your own method, and then used it for identification of the interactions. Very interesting lecture. I really liked that very much.
Thank you, sir.
Thank you. Thank you very much. Thank you very much.
So any student
or any faculty, they can share your feedback or suggestion to make it better, to make it more reachable to the society. This talk should go to more people. So you can suggest. क्योंकि हम समझते हैं साइंस में आर्गुमेंट एंड काउंटर आर्गुमेंट इज रिक्वायर्ड फॉर इम्प्रूवमेंट. समटाइम्स वी मिस समथिंग, वी कीप ऑन आस्किंग द क्वेश्चंस.
You can ask any question. I will try to answer.
Yes.
Yes. I have one. Professor Mishra, you gave some.
Sir, please call me by my first name. You are my teacher.
Okay.
Please don't say that.
Neeraj, you mentioned some site some link where we can get all those high resolution pictures, very colorful pictures of the cell which you showed.
Yeah.
Can you write down that in the chat box so that
that is PDB site, sir. PDB.
Oh, I see.
PDB site.
Protein databases.
Yes, sir. Now they have changed the name to RCSB. But if you will type PDB, it will come.
Okay. Fine. Got that point.
Yes.
Thank you.
So
No
student
question.
could share feedback. Varsha, Janab, few people are joining now. 45 मिनट के बाद लोगों को पता है feedback देते हैं, so they join after. So sometimes I give earlier. So now you have seen 2-3 people are asking for feedback, but they have joined after 45 and 6:45, 5:45. Sorry. So they are not able to see in chat box. So you should try to learn. Your intention should not be to get 1 hours का आपको सर्टिफिकेट मिल भी जाएगा तो कोई ज्यादा फायदा नहीं होगा. We will give you certificate, but intention is, our motto is to learn. Some things not 100% तो हमको भी नहीं समझ में आता है, but at least 10%, 20% it will help you gradually. And
The point is, Gopalji, this is part of chemical biology. Because this is not natural amino acid. Fluorine is not found in cell. There is no biological system can incorporate. So I can understand this is more chemistry, physical chemistry. That's why people are not coming in biology to
entire body is composed of chemical, sir. Entire body is composed of chemical. You know कि I take 4-5 hour lecture on chemistry and mathematics for biologists. So I have seen कि even MSc student they are not able to calculate polarity, normality. They are not knowing. See, still people are joining. They know कि 5 p.m. is time. See, Fatima Nazim, I am allowing her. Welcome, Fatima. You have joined on time. We are going to close this lecture. Fatima. We are going to close this, wind up this lecture. But welcome, you are welcome in this session. If you have any suggestion, please share your suggestion. Then we will wind up. So there is no suggestion. Then good night, everybody. Thank you, Poonam Mishra, madam.
Thank you, madam.
Sir, Neeraj Mishra, sir.
Thank you, sir. Thank you, Virdi, sir.
Thank you very much.
सर आ जाते तो अच्छा हो जाता है ना, इम्पैक्ट बढ़ जाता हमारे लेक्चर का, है ना. सर आप भी एक बार आई रिक्वेस्ट यू ओपनली कि यू शुड आल्सो टेक, आपका जितना एक्सपीरियंस वाला एक दिन पूरा कहानी 1 घंटे का साइंस में बताइए सर.
हाँ डेफिनेटली बताएंगे, डेफिनेटली.
यस सर, दैट विल बी गुड.
Yes.
So good night.
Good night,
everyone.
Thank you so much, sir.
Again we will meet on 21st. There are 2 lectures on 21st.
Okay.
Okay.
Okay, sir.